Which algorithm is most commonly used for multiple sequence alignment in bioinformatics? A. Needleman-Wunsch. B. Smith-Waterman. C. BLAST. D. CLUSTAL-W. The correct answer is D. CLUSTAL-W is widely used for multiple sequence alignment, optimizing the alignment of multiple sequences by generating a guide tree and progressively aligning sequences. In a phylogenetic tree, what does a node represent? A. A common ancestor. B. A mutation site. C. A specific gene sequence. D. A branch length. The correct answer is A. A node in a phylogenetic tree represents a common ancestor from which two or more descendant tags are evolved. Which of the following is a database specifically designed for protein-protein interaction data? A. PDB. B. String. C. GenBank. D. Ensemble. The correct answer is B. String is a database that stores known and predicted protein-protein interactions, integrating both experimental data and computational predictions. Which of the following best describes a hidden Markov model, HM, in bioinformatics? A. A sequence alignment algorithm. B. A statistical model used to predict protein secondary structure. C. A probabilistic model for analyzing biological sequences. D. A tool for visualizing phylogenetic trees. The correct answer is C. Hidden Markov models, HMMs, are probabilistic models used in bioinformatics to identify patterns in biological sequences, particularly in gene prediction and protein family classification. What does the A value represent in the context of a BLAST search? A. The evolutionary distance between two sequences. B. The number of expected matches by random chance. C. The percentage identity between two sequences. D. The gap penalty score. The correct answer is B. The A value in BLAST represents the number of alignments with a score equivalent to or better than the given match expected by random chance. A lower A value indicates a more significant match. Which file format is typically used for storing nucleotide or protein sequences in a simple, readable text format? A. FASTQ. B. GenBank. C. PDB. D. GFF. The correct answer is A. The FASTQ format is a widely used file format for representing nucleotide or protein sequences, consisting of a description line followed by the sequence data. Which of the following tools is used for aligning two nucleotide sequences based on global alignment? A. BLAST. B. Needleman-Wunsch. C. Smith-Waterman. D. MAFFT. The correct answer is B. The Needleman-Wunsch algorithm is designed for global sequence alignment, aligning sequences from end to end, ensuring the best overall match. Which bioinformatics tool is commonly used for gene expression analysis by aligning RNA-Seq reads to a reference genome? A. BLAST. B. Bowtie. C. MEGA. D. MAFFT. The correct answer is B. Bowtie is a fast and memory-efficient tool used for aligning short RNA-Seq reads to large genomes, commonly used in gene expression analysis. What is the primary function of the Gene Ontology (GO) database in bioinformatics? A. Storing protein structures. B. Describing gene functions. C. Predicting protein interactions. D. Modeling metabolic pathways. The correct answer is B. The Gene Ontology (GO) database is used to describe gene products in terms of their associated biological processes, cellular components, and molecular functions. In the context of next-generation sequencing, NGS, what is the purpose of read mapping? A. Predicting protein folding. B. Aligning sequencing reads to a reference genome. C. Creating a phylogenetic tree. D. Annotating gene functions. The correct answer is B. Read mapping involves aligning sequencing reads from next-generation sequencing, NGS, experiments to a reference genome to determine the origin of each read and to identify variants. Which of the following describes the process of sequence annotation in bioinformatics? A. Aligning sequences using multiple alignment tools. B. Predicting the 3D structure of a protein. C. Identifying and labeling functional elements in a sequence. D. Building a phylogenetic tree. The correct answer is C. Sequence annotation involves identifying functional elements in a DNA or protein sequence, such as genes, exons, regulatory regions, or binding sites. What is the key distinction between de novo and reference-based genome assembly? A. De novo assembly uses known reference genomes to build a sequence. B. Reference-based assembly relies on aligning reads to a known genome, while de novo assembly constructs the genome from scratch. C. De novo assembly is faster than reference-based assembly. D. Reference-based assembly uses longer sequencing reads. The correct answer is B. In de novo assembly, genomes are constructed from short sequencing reads without a reference, while reference-based assembly aligns reads to an existing genome for sequencing. Which of the following terms describes the process of comparing a query sequence against a sequence database to identify similar sequences? A. Homology modeling. B. Sequence annotation. C. Sequence alignment. D. Database search. The correct answer is D. Database searching involves comparing a query sequence to a database of known sequences, EG, BLAST, to identify homologous or similar sequences. Which of the following is a widely used software for constructing phylogenetic trees from aligned sequence data? A. BLAST. B. MAFFT. C. MEGA. D. Clustal Omega. The correct answer is C. MEGA is a comprehensive tool used to analyze sequence data, construct phylogenetic trees, and perform evolutionary analyses on aligned sequences. In the context of protein structure prediction, what is homology modeling? A. Predicting protein structure from scratch. B. Constructing a protein model based on its homologous proteins with known structures. C. Using sequence alignments to predict secondary structures. D. Aligning sequences based on functional domains. The correct answer is B. Homology modeling predicts the 3D structure of a protein by aligning it with homologous proteins that have known structures and using those structures as templates. Which of the following bioinformatics techniques is used to identify evolutionary conserved regions in sequences across species? A. Multiple sequence alignment. B. Phylogenetic tree construction. C. Genome annotation. D. Protein folding prediction. The correct answer is A. Multiple sequence alignment is used to align sequences from different species, allowing identification of evolutionarily conserved regions that may indicate functional or structural importance. Which of the following methods is used to predict gene structure from RNA sequencing data? A. Homology modeling. B. Sequence annotation. C. Transcriptome assembly. D. Read mapping. The correct answer is C. Transcriptome assembly from RNA sequencing data is used to reconstruct the full-length transcripts, thereby helping predict the structure of expressed genes. What is the primary goal of genome-wide association studies, GWAS? A. To study protein-protein interactions. B. To compare genomes of different species. C. To identify genetic variants associated with diseases. D. To assemble complete genomes from short reads. The correct answer is C. GWAS aim to identify genetic variants, typically single nucleotide polymorphisms, SNPs, that are associated with traits or diseases by comparing genetic data from affected and unaffected individuals. Which of the following describes a FASTQ file used in bioinformatics? A. It stores aligned sequence reads. B. It contains nucleotide sequences along with corresponding quality scores. C. It is used for storing protein structures. D. It contains phylogenetic tree data. The correct answer is B. A FASTQ file stores both the nucleotide sequence and the corresponding quality scores from sequencing reads, typically generated in high-throughput sequencing experiments. Which tool is commonly used for identifying conserved domains in protein sequences? A. PSIBLAST. B. BLAT. C. HMMER. D. Clustal Omega. The correct answer is C. HMMER uses Hidden Markov Models, HMMs, to identify conserved domains in protein sequences, which are crucial for predicting functional and structural regions.